Cell Imaging Core is a centralized facility where users receive technical support to do all sorts of imaging and flow cytometry. Pretty much all fields of biological sciences really need imaging and flow cytometry these days. It's really important for cancer research, for immunology, for neurobiology research, for any molecular studies where they're trying to investigate mechanism towards better understanding of disease, but also just basic understanding of physiology. You cannot do those studies without imaging and flow cytometry. We provide service at all levels. So young researchers that have never worked with microscopes or flow cytometers previously can come. They will be taught how to use the instruments themselves, and it's part of their training as researchers. We will also help them in study design and in data analysis. Currently, we train people to use rather powerful imaging software, such as BioImage XD, also ImageJ, and Immerse. And people like to learn how to do the image analysis and visualization themselves, but some customers want a full service. Very often the companies especially, they want to be hands-off. They even want us to design experiments for them, and then we take the samples, we do the imaging or the cell sorting, whatever the case is, the analysis, and basically they get the full package, and we're very flexible. We always strive to keep up to date with new technologies. That's really important. But in particular, we are developing our know-how, I would say, in the area of diagnostics and drug discovery. So for imaging or microscopy, this means high-content imaging. For flow cytometry, technically it means getting new hardware, but there's a lot of new diagnostic assays being developed using flow cytometry, and this is in the field of cancer and immunology in particular. The spatial information that you get from images just opens up a new view to this world, and many problems may not have been solved without this spatial information. You also get temporal information from imaging. Flow cytometry is spectacular because of the single cell analysis potential. Well, it already is used for single cell analysis, but the single cell technologies in general are developing a lot, and also in the area of flow. So we now appreciate that there's a lot of heterogeneity between cells in terms of their signaling, and sometimes this is important to understand. But also the whole area of drug discovery, really needs high content imaging. So not just looking at lots of samples, but looking at many features in those samples and looking at those features over time. So it opens up new horizons.